Hello and welcome to this week's What Were They Thinking? We begin with an article coming out of the Bloomberg publications talking about the long-term effects and that even though a number of patients are clinically treated and cured of the CCP virus, they continue to exhibit side effects of the infection. These include effects in the eyes, nose, throat, heart, lungs, muscles. There's more that goes on. These symptoms are exhibited for months after treatment has been finished. This is not the first time such long-term consequences have been reported, and it's likely to be part of a long-term, ongoing body of evidence that the CCP virus isn't only an acute issue, but may have generated a number of chronic issues in patients, both those who were severely sick with it and those who were not severely sick. Consider the millions of Americans alone and the nearly 500 million worldwide who have been infected by the CCP virus. If even a small fraction of these were to have long-term chronic illness as a consequence, we're talking some serious medical overflow issues that are just going to exacerbate what we're already seeing. The purported silver bullet to all of this is a vaccine, and there is every reason in the world to believe a vaccine would be effective. The concerns at the moment are largely about what the vaccine is made from, where it's coming from, and whether or not it's safe. This last element about safety is also being compounded, let's say, by the testing in a largely adult population. There is little to no evidence of its efficacy or safety in children, and there could be some legitimate concerns about giving a vaccine like this to kids, given how little we know about how safe it is based on these relatively large but overall very small trials that were run in record time. That may not be an issue immediately, as initial rollout of any vaccine is likely to be delayed by the simple fact that we need to get enough of a stockpile and then distribute it to adults, let alone focusing on children. As one of the sources for this information has stated, they did contact several of the drug companies that have front-runner vaccines, and they have said they plan to test it clinically in children, but there is no timeline for this. This also makes sense. If you have a drug and you can establish safety in adults, then it is that much easier to establish safety in children. However, running two parallel clinical trials where you are uncertain of the dosing regime for either is simply doubling the problems you have on hand and possibly causing harm to the minors involved. In other news, as it has been reported by the media, the CDC reversed the guidelines it put out that testing should be limited to only those with symptoms. This was, at least at the time, in contrast to the World Health Organization, which held that you should be testing as many people as possible. The headlines often read something to the effect of that the CDC has reverted back to guidelines that anybody who has either been in contact with or could have been in contact with a CCP virus carrier should be tested. According to some sources, there is a claim that the guidelines that were issued and then reverted from were not actually made by the CDC, but instead created by the Department of Health and Human Services, and thereby circumvented the CDC's normal process for ensuring information on its website would be accurate and properly vetted. This information further claims that the new guidelines were given despite opposition from the CDC itself. Hopefully, with the reversion of sorts, the new testing guidelines should allow for a large portion of the population who otherwise are asymptomatic but carriers of the CCP virus to be detected, engage in appropriate healthy behaviors such as isolation, and thereby begin reducing the breakout numbers yet again. To highlight just how serious the CCP virus could be in the long term, we want to talk about an article coming out that's discussing the possible relationship between CCP virus infections and Parkinson's disease. We've previously described that there are definitely neurological symptoms associated with being infected by the CCP virus. This is because it appears to be able to make access to the brain, and from here, cause damage. The body then has to react to this, and this is where a new study comes in. It posits that the next wave of the CCP virus may not be a third wave of infections, but a third wave of conditions given rise by the presence of the CCP virus, specifically Parkinson's disease. They believe that inflammation in the brain could be triggered by the virus presence and the immune response. They base their theory on historical evidence, specifically evidence coming from the 1918 Spanish influenza outbreak. At the time, they were referring to something called encephalitis lethargica, and this was directly related to the pandemic itself, and that this then increased the risk for Parkinson's by 2 to 3-fold. This whole situation, both now and in the future, could become a much bigger issue than it currently is, and for this reason, among others, the World Health Organization has finally agreed to allow testing of traditional herbal medicines against the CCP virus. Although, as Tim Minchin once put it, medicine that works is otherwise known as medicine, and traditional herbal medicines historically don't have the same degree of value. In some instances, they have niche applications, and on occasion, very specific but otherwise trace amounts of important and useful compounds can be identified from them. In other instances, the raw, unrefined form of the herbal medicine is not particularly practical, and therefore bioprospecting companies have been able to take these, modify them, and create new medicines. In fact, historically, this is how we get most of our malaria treatments. The World Health Organization's endorsement of this as a viable avenue of investigation is both relatively moot if we have a successful vaccine, but also something that we have largely investigated. The large libraries of information containing the structures and nature of many of these compounds have been sifted through to try and identify what could possibly be used as a treatment or vaccine for the CCP virus. That the World Health Organization would agree to allow various organizations to enter into Phase 3 clinical trials for herbal medicines would indicate just how desperate they are to try and get the CCP virus under control. Trace amounts of anything can, at least in theory, be picked up and then used, and this is exactly what's being seen at Helsinki. The Helsinki Airport is using sniffer dogs to find patients who have the CCP virus who don't know they have the CCP virus. A dog's sense of smell is significantly better than a human's, amazingly so by comparison, which is one reason why they're used to find hidden drugs. That degree of detection is also useful for identifying other things. There have been attempts to see whether or not we could train dogs to detect cancer in patients, with varying degrees of success. The idea of using a similar approach to identify the CCP virus carrying patients is not that far-fetched. Various studies have already shown between a 94 and 95% accuracy rating for dogs using either saliva or sweat. Although they are not perfectly accurate, what you gain in lieu of that accuracy is speed. A simple swab, a couple of minutes for a dog to sniff it and be able to give an indication as to what it thinks is there, is a lot quicker than shipping off nucleic acids to a lab, waiting for the test to be completed, and then receiving results back. This may take several hours. Even the quicker tests are often expensive, relative to the ongoing expense of maintaining a dog. Going now from the CCP virus, we want to talk about the wild, but more of wildlife than something as dangerous as the CCP virus. In America, there are a number of issues that are simmering under the surface, ranging everywhere from political unrest to the Yellowstone volcano. One specific issue relating to animals is the presence of feral hogs, or wild pigs, however you happen to know them. These are pigs that have gone rogue, developed in the wild, and, as a result, lost many of their desirable temperaments and become far more aggressive. This is also one reason why they cause a lot of damage every year. The downside to this is that they're also as smart as a regular pig. Most pigs are quite intelligent, and as a result, the wild pigs are more dangerous for it. They also have a good sense of smell, and unlike the dogs being trained to sniff out the CCP virus in the airport, this is often used to hunt down food, small animals, and, in certain countries, truffles. Wild pigs are also much like a boar, and boars historically have been very dangerous to hunt. This was because of their large tusks and very dense heavy fur and hide. This meant it was hard to kill them or injure them. Finally, the biggest issue of all is that they are an introduced species, but more than just being an introduced species that has proven itself surprisingly versatile, they've been bred to be continuously fertile, and to have large litters. This means, like rabbits, foxes, and mice, they breed very quickly and in large numbers, can feed on many things, and ultimately take over certain biomes and ecologies. This has led to the current estimate of over 9 million feral pigs in the United States of America alone. This problem is likely to continue growing over the coming years. While we're talking about animals, the pigs may be growing out of control. However, a rare and endangered species known as the Javan rhino appears to be coming back, or at least is possibly coming back, after a mother and calf were recently seen. These two are part of the total number of 74 Javan rhinos. These animals live in roughly 12,600 acres of rainforest, and the government is going to a lot of effort to try and maintain the species. Next, we have some closure to the mysterious die-off of elephants in Botswana. It was thought that over 300 of the elephants in Botswana had died mysteriously, going back as early as March. We know it wasn't poachers because their tusks were still there along with the meat and hide. This means there weren't any hunters involved either. The fact that such a large animal had died mysteriously, but often in proximity to water, led authorities to think that it was likely that they had either been poisoned or come into contact with some sort of pathogen that lived in the water. What appears to have been happening is the elephants were poisoned by cyanobacteria. This was a waterborne pathogen that was sitting in stagnant water in relatively large pools that the elephants were drinking. When they drank this, they were also consuming a neurotoxin generated by the cyanobacteria. This was then found in the elephants' bodies when blood was drawn. The fact that the elephants have stopped dying off coming into June, along with the drying of these bodies of water, would indicate that there is a close association with it. It's fortunate because it's ruled out several other factors that would have been more concerning, these being particularly anthrax or some particular form of human poaching. While on the subject of bizarre behaviors and microbes, the fungus that gave rise to penicillin as we know it has finally had its genome sequenced. Penicillin has what could be considered an apocryphal history behind it. It is said that Alexander Fleming had a piece of bread growing in an agar plate. It developed mold on it, and around the mold no bacteria would grow in that agar plate. And so he decided to test what was happening and see if it could be utilized somehow. This eventually led to the discovery and production of the most well-known antibiotic family, penicillin. The very first usable strain of the penicillin mold was frozen and preserved long ago. A small sample has since been taken out and its genome sequenced, and this is helping in understanding not only what's going on with the mold, but also the production of penicillin. The genome sequencing looked at both the original Alexander Fleming penicillin mold and a strain developed in America. The American strain was somewhat unique by comparison in that it was created through irradiation and a variety of other, very crude genetic modification techniques, and then selected for its ability to produce antibiotics. This has led, at least in the findings from this study, to significantly more copies of those genes involved in producing penicillin in the American strain than in the original Fleming strain. Antibiotics are, at least in theory, one of the go-to treatments for many diseases and pathogens. One of the diseases it does not work for is malaria. Malaria is a parasite, and for this reason, it's not an effective treatment, much as antibiotics are not effective for viruses. What is unusual is that some people have, for lack of a better word, an unusual mutation in their blood that can help them ward off malaria. This is even better than what some vaccines can achieve. The genetic mutation responsible for this is, at least, relatively isolated in where it occurs, primarily in a small population within Africa itself. The researchers explain that the way this particular mutation works is that it increases the tension of the red blood cell's surface, and that this causes it to have difficulty accessing it, despite having all the means available. That surface tension is simply too much for the malaria parasite to overcome. Having one copy of this gene would give 40% resistance to the malaria parasite. Having 2 copies was equivalent to having 74% protection. At least as far as the research goes, this is often compared to sickle cell anemia. Sickle cell anemia, if you carry 2 copies, causes all the red blood cells in your body to become mutated and not function properly and then be destroyed by the immune system. This, if you pick up the malaria parasite, will obviously destroy the malaria parasite along with the red blood cells that they've infected. If you carry just 1 copy of the sickle cell anemia gene, some of your red blood cells will be destroyed, along with some of the malaria parasite, but you'll remain relatively healthy. If you carry no copy of the sickle cell anemia gene, then all of your red blood cells are vulnerable to being infected, but your body does not destroy them through the immune system. And so this has led to the bizarre phenomena in parts of Africa where those people who carry a slightly adverse gene are actually at an advantage in society as they are healthier, live longer, and consequently produce more offspring. The fact that a mutation exists that achieves the same outcomes or very close to without the adverse effects of having the immune system destroying some of the red blood cells in the body is incredibly desirable and could, at least in theory, lead to ways that we could manipulate the human body via drugs to be more resistant to the malaria parasite. While we're on the subject of diseases, we should talk about diabetes and a possible new device coming from Japan that could help you check your blood glucose levels painlessly. At the moment, there are really only a handful of ways to try and check blood glucose levels, these being whole blood tests, which are time-consuming and tedious, and often unnecessary. Then you have the prick test, which involves taking a small sample of blood from a finger, placing this on a strip, putting the strip into a blood glucose measuring device, and measuring it that way, or having real-time measures through your infusion device depending on what type of diabetes you have. For the most part, all of these involve some degree of discomfort. This device itself is designed to have extremely tiny needles that can enter the human skin but not be noticeable due to their size. This would allow for easy testing. Unlike the current measures which require getting it through as much of the skin as possible, down to the capillaries where blood will then come out, these microneedles on the device allow it to get to just below the surface of the skin, where it can take a measure of the glucose levels, but not so deep as to interfere or activate the sensory neurons. This means you don't sense its activity and presence. The advantage of this particular approach versus having to use needles is that it is low cost, disposable, and most importantly does not require training or run anywhere near the same risk of infection. Understand that anything that involves penetrating deep enough into the skin to draw blood always carries certain risks. If you don't go as far as the capillary bed and therefore drawing blood, that risk is substantially reduced. It would be a very localized infection, and the body is far better adapted at dealing with these versus something getting into the blood supply of the body. The reason this works for glucose measuring in particular is that glucose enters into the interstitial fluid around the cells, and then eventually the cells take it up. What this device does is measure it before it goes into the cells, giving a good indication of just how much glucose is present. The practical applications of this, above and beyond glucose, are things like drugs. Rather than having to rely on taking blood for law enforcement, it is possible for them to use one of these tests. Instead of having to take blood, simply placing it on the skin and looking for a color change, much like what is done with disposable test kits for drugs found as a whole product. Those are all the news items we have for you this week. Hopefully you've found them interesting. Thank you for watching this video. If you've enjoyed it, consider liking, sharing, and subscribing. Please post any comments, questions, or suggestions below.